So I guess we're good to go. A little bit late starting, but that's all right. So we're doing chapter 11 today. So we're going to start talking about body senses and movement. And so for today's class, we're mostly just going to do the body senses side of things. And then we'll sort of switch over to talking about movement for Thursday. But when we talk about our body senses, there's a couple of different things we can refer to. We can be talking about proprioception or proprioception. I can never figure out where to put the emphasis. Or we can call it kinesthesia, which is how I usually would refer to it. But these are our sort of way of sensing movement when we're having sort of actions in the location of our body. That would be our proprioception. We also have our skin senses. So these are going to sense the conditions at the surface of our body. So this would be sort of heat and cold and pain and pressure. All of those are skin senses. We also have our vestibular system, and this involves our sort of inner ear, and it helps us figure out our orientation and our body position and whether we're moving in space. And the last one is our interoceptive system. And this is just sort of being able to introspect and sense what's going on inside our own bodies. So that's going to be the states of our internal organs. And that last system is mostly going to be in the background. It doesn't really overlap a whole heck of a lot with behavior. So we spend most of our time on the other three systems. We just mention the fact that we have an interoceptive system, and then we can move on from it. But we're going to talk just a little bit more about each of these, and they get at least a slide each, which is kind of nice. For proprioception, this is the sense that informs us about the position and movement of our limbs and body. So this is going to be receptors that are in our joints, things that are going to detect how much our muscles are sort of bent or not bent. And they're very key to a lot of our basic functions. So things like maintaining our posture and intentionally moving our limbs. It also helps us with grasping and locating objects. So, as I said, we have sort of sensors that are going to report the tension and how long our muscles are stretched out. They're going to then be able to sort of infer joint angles. So is our arm straight or bent? What are we doing with our fingers? Your body's going to track all of those things. And it also tracks the head as we move around in our world. So as you turn your head, as you look to different places, this is going to keep track of sort of which way you're facing and how you're moving. And then if you think of it, say every morning you always put your phone right beside you on the bedside table, and it goes, or your alarm goes off in the morning and you reach for it, you don't necessarily even have to look for it, you just know where it is. Or maybe you put down a pen to pick up a pencil, and then you want to switch back so you can reach to where you left the pen and grab that instead. This system would sort of help you keep track of those things so you don't have to visually locate things. You can also have sort of a sensory memory to it as well. And then there are some special cases where individuals can have damage to this system. And if you don't have proprioception, you can have sort of a lack of bodily control. So you might not be able to maintain tension. So instead of having sort of a vertical upright posture, maybe you flop over a little bit and have trouble supporting yourself. And in some of the cases where people have had this, usually from some kind of infection that affects the nervous system, they require years and years of specialized training with sort of reincorporation of visual senses to be able to account for and do all of the things that this system does naturally in the background. So if they can't see, they aren't able to maintain that proper tension and might just sort of have a full body collapse, which is really, really interesting. And then there are certain genes that seem to be related to this system. So if you have mutations in particular genes, you might have more likelihood or an increased likelihood of deficits in this proprioception. So it's neat to see that it also has a little bit of a genetic component, but can also be caused purely by things like environmental influences as well. All right, so next we can talk about our skin senses. And we're actually going to go into more detail in the skin senses than we did with the proprioception. So our skin senses, as I said earlier, deal with warmth and touch and cold and temperature and pain. So there's a lot of different things that we can sense, so lots of different sensations. Some people will argue for the inclusion of sort of tickling and itching sensations, but that's another one of those areas that are up for debate. But each receptor will have sort of its own type of sensory information that it detects, and they'll also have their own unique pathways from those receptors to the brain. And we could actually call those pathways a labeled line, where we know exactly what pathway it's going to take, and we know what kind of information it's bringing straight from the surface of the skin all the way up to the parts of the brain that actually interpret that information. And the textbook actually used a really interesting way of showing these labeled lines and the sort of distinction between different areas. So they recommend that if you have, say, a pencil with a lead tip, and if you run it along your skin, specifically probably your hands or your face, because that's where you have the most receptors, you'll feel the touch of the pencil continuously, so you'll be able to track its movement. But the fact that the pencil's a little bit colder than your skin, it will only feel cold in certain places. So we only have those cold receptors in very distinct locations, and everywhere else we're just getting that tactile sensation. And so they're separate, and they run as sort of independent systems, giving us that feedback. But it's kind of neat. I didn't think that it was that simple to feel the difference between sort of tactile sensations and temperature sensations, but apparently you can. All right. And so we're going to talk about a whole bunch of different types of skin sense receptors. So as you might imagine, if we're looking at things like tactile sensation and hot and cold and pain and all of those other things, there are definitely going to be a couple of different kinds of receptors. We're also going to end up having a little bit of almost redundancy, where we have multiple types of receptors that will give us the same kind of information. So we can have multiple temperature sensors, and it sort of just depends on what they're looking for and how they react, because they're going to be informing different parts of the brain. But our two big types or big categories of these receptors are either free nerve endings or encapsulated receptors. And this basically means that if you have free nerve endings, they're going to have sort of processes that are existing just at the ends of a dendrite. So this is usually where we see temperature perception, so warmth, cold, as well as pain. And this just means that they aren't sort of a specialized cell type that's at the end. If we think about our vision, our sensory receptors are those rods and cones, some kind of specialized cell that responds to certain stimuli. This is just a normal neuron that has receptors that can pick up on this information. And we'll talk about some of those receptors. Our encapsulated receptors are a lot more similar to what we know from the other bodily senses, or I guess other senses in general. So they are going to be more complex structures that are enclosed in a membrane. So typically standalone cells that are going to have some kind of function. And so instead of doing the warmth and cold and pain, these typically detect things like touch. And it gets a lot more specific than that. We can look at shape and texture and size and location and all of those other things as well. So we'll break those down in just a minute here. And for those who are visual learners, we can look at this sort of cross-section of the skin, and we can see a couple of our encapsulated receptors. So we have Merkel's discs, Pacinian. Oh my goodness, I was trying very hard not to say Inian. I don't know why. We have the Ruffini endings and Meissner's corpuscles. So you can see that we have sort of two that are up higher in the surface level of the skin. So the Meissner's corpuscles and Ruffini's endings, those are up in sort of an upper layer of the skin. And the Merkel's discs and Pacinian corpuscles are lower down. So just in different layers of the skin. We don't care too much about the distinct layers. We're just sort of making note that that's where they're sitting. And then our free nerve endings are typically right at the surface. So because they're interacting with what's going on at the surface of our skin, they're sitting just at that outer layer of our skin cells. And so you can kind of see those guys in there as well. And like we said, they're just the ends of neurons. So they're just the dendrites that are there, ready to be stimulated. So they just kind of hang out just below the surface. So we're going to be talking about each of these types and what they're good at a little bit, in a little bit more depth, but very, very briefly for what we could talk about them. So for our specifically encapsulated receptors, these are the Meissner's corpuscles and Merkel's discs. These are the ones that are sort of in the mid layers, not very deep in the skin. And they tend to detect texture and fine detail, which kind of makes sense. The closer you are to the outside of the skin, the better you are going to be able to get fine details and textures of things that are touching the skin. And the difference here is that the Meissner's corpuscles are going to have sort of brief, very, very quick responses. So we'll have signals sent to the brain very, very quickly and very short in duration. Whereas the Merkel's discs are going to be a more sustained response. And then our brain interprets that pattern of firing from those two distinct types of receptors and interprets the texture and the detail on whatever it is that we're touching. And then for our deeper layers, we have our Pacinian corpuscles and Ruffini endings. Those are much deeper down, and these detect stretching of the skin. So instead of being close enough to the surface to be getting more fine details, they're focusing on, okay, well, how is the skin changing? What's going on at a larger, sort of more macro level? And so this helps us perceive the shape of objects that we're interacting with. So especially if we're grasping something, so if you're holding something in your hand, we can kind of feel how our skin is moving to encapsulate that thing that we're holding. And then these receptors are involved in that. Okay, and then the next ones I want to talk about are our free nerve endings. And for these, we're going to be sort of dividing them up based on what it is that they're detecting. As I said before, we have sort of temperature and pain and different things like that that we can get from these free nerve endings. And there are many, many, many different types of these. And most people won't actually agree on exactly how many there are. So we're just going to talk in general terms here. But for our temperature receptors, all of the identified temperature receptors seem to be sort of members of a transient receptor potential, or TRP, family of protein ion channels. So whether they're detecting hot or whether they're detecting cold, because they're not going to do both, they're going to respond to one or the other. They're basically going to be receptors on sort of, if we think back to the discussion about how ions move into and out of our cells, these would be protein ion channels that are specifically going to only bind when particular conditions are met. So if it's too hot, the ones that detect heat will bind with whatever it is that they're detecting there and open up, allowing an impulse to be created. Or maybe they only respond to cold, so maybe they change conformation due to the energy that isn't present, because cold would be sort of a lack of extra energy, and then that would sort of change that influx of ions, again, in a detectable way. So there's a bunch of different ones that respond to sort of different intensities of temperature. And there's some focus on heat, some focus on cold, but they all seem to go about doing it by controlling the influx of ions into our neurons, again, in a predictable pattern that our brain can then interpret as hot or cold, and specifically in amount of hot or cold. For our pain receptors, we have 3 distinct types of pain. So it's not just a simple, this hurts. We have thermal and chemical and mechanical pains. So the thermal goes back to our temperature receptors, where if we are too hot or too cold, we can feel pain. So if you touch a hot pot that just came off the oven, that's going to give you thermal pain because it is too hot and it's burning your hands. And if it's too cold, so maybe you go outside today without a coat on and your skin starts to freeze. So that would be sort of too cold, and that would cause pain as well, but for a different reason. And so those are still going to be the TRP receptors, and they're going to tell us sort of what's going on. Though the textbook does take a moment to note that the sort of painful heat is just like our normal temperature reception for warmth, but our painful sort of threshold for cold, they have no idea how that works. So since the textbook has been published, there might have been more studies, but what that usually means is that they have lots of different proposed theories, and they disagree with one another. So they don't quite understand why being too cold hurts, but it doesn't use the same setup as our normal temperature reception, which is kind of cool. Our second kind of pain is chemical pain. So this is sort of, again, a completely different category, but it's looking at different kinds of chemical irritants, so things that react with our skin. So if you think of something like capsaicin in hot peppers, that's going to cause sort of a heat pain. So that would be a chemical receptor responding to that because it's binding to a chemical in that particular item. We also have the coolness of mint and menthol. That would be a coolness receptor, and it's again responding to chemicals that are producing that sensation. So it's not actual hot or cold, it's sort of perceived hot and cold. Mm-hmm. I can't quite remember. I think what it does is it makes it harder for it to bind to the receptors. So if we think of like our taste buds and things getting into those grooves where they can bind to our different receptors, I think the milk just sort of keeps them away from it, but I'm not positive on that one. Because I don't think it's like a physical reaction where they interact with each other and like disable each other or anything like that. I do think it's more of a keeps it away and creates a barrier, but I'm not positive. All right, and then the last one are mechanical pain receptors. So this is when we have sort of physical sensations of pain. So maybe something has hit too hard or something has damaged your skin. That would all be mechanical kinds of receptors. And again, we run into this issue where they really don't understand what is driving mechanical pain reception. So their understanding of the receptors here is very, very poor. And that's really unfortunate because there are a lot of pain issues, say people with hypersensitivity, where even small levels of mechanical stimulation lead to severe pain. But they don't really understand what's going on there. So again, probably a lot of different conflicting reports of what's happening, but no consensus in that field, which is, again, very, very interesting. Okay, next we're going to talk about our vestibular sense, and at the beginning I kind of mentioned that this has to do with our inner ear. So this helps us maintain balance. It's going to provide information about our head position and if we're moving or not. And it's maintained by our fluid-filled vestibular organs in the inner ear. So if you remember, back to sort of looking at our sense of hearing, we also have these semicircular canals. And so there are 3 of them, and they have different orientations. And you can kind of think of them as being on different axes. So you have one that's in a horizontal plane, one that's in a vertical plane, and then one that's sort of on a diagonal. And we can interpret the movement of fluid in those 3 different planes and figure out, okay, well, if gravity is always pulling down, what's the fluid doing? Where is down based on where our head is located? And so within those semicircular canals, there's fluids that move around, and it's going to brush against hair cells that react to that movement. So we can detect where that fluid is based on sort of what it's pushing up against. Okay. And then I think I've mentioned this before, but it's hard to remember which classes I have and haven't said this in. But if you do something like sit on a spinning desk chair and you spin and spin and spin and spin, at first the fluid will start sloshing around because you've started moving. So you've started that movement and your brain is going to detect that movement. But after a while, if you spin consistently, the fluid's going to stop sloshing because you're moving fairly consistently. And then when you stop moving, it's going to slosh again. But your eyes are telling you that you're not moving, but your ears are responding as though you're still moving because of that movement in the fluid. And that sort of contradiction in your different senses of movement is what makes you feel dizzy, because your eyes are telling you that you're stationary, but your ears are telling you that you're moving. I think this also has something to do with why some people get sick in a car. So if you think about it again, sitting in a car, you are moving, but once you get up to speed, you're not really accelerating. You're kind of at rest. So even though you're moving forward, the fluid isn't going to be moving because it's consistent motion. But your eyes are seeing movement. So as the outside moves, you would sort of assume that you should be moving, but you're not. And that, again, disconnect between what our ears are telling us and what our eyes are telling us can lead to some inner system problems. So some people get really nauseous, some people are going to throw up, some people just get dizzy. But again, we have sort of these different systems that let us know what's going on, and if they disagree with one another, then we can run into some issues. All right. So next we're going to start talking about, okay, so this information is coming from all of these different parts of the body. Where is it going to be sent? And so specifically looking at things like on the surface of our skin, where's that information going to be sent? And how is it going to be sent? Because we've mentioned that the brain is able to interpret what's going on based on sort of this base knowledge of where things are coming from and what they mean. So the first important term here is the idea of a dermatome. And this is a segment of the body that is served by a single spinal nerve. So if you remember when we were looking at all of our different spinal nerves, they kind of leave in between different vertebrae. And so they're going to reach out and serve the rest of our body. And so we actually can see, okay, which parts of our skin will feed to this nerve versus that nerve versus the one above it or below it. And so we can actually see that we have almost like distinct bands around our body that show us which parts of our skin is fed through which of our nerves. And it's a little bit misleading here. So this diagram makes it look like we have very distinct separate bands of detection. But there is a lot of overlap between these bands. So this sort of shows us the average, but there is an overlap of as much as half of the parts of the skin. So if we have something right along the line, say between T3 and T4, we might say that, okay, well, the stuff that's in the middle is detected by both T3 and T4. And that allows us to get some redundancies. And so it would allow us to sort of detect stuff as it moves between zones and keep track of it. It also does a really great job of allowing us to compensate in case of damage to one of those nerves, because the redundancy can help cover some of the lost function. So it's an evolutionarily beneficial setup, even if it does end up having us sort of using more resources than we typically would need to. It can be beneficial if we're injured. All right, and so like we've kind of been saying, we've got this body sense information that's coming into our spinal cord through those spinal nerves, or if we're up high enough, it can be coming into the brain directly through cranial nerves. So it's entering into our central nervous system somehow. And typically the information is going to cross over our midline at the medulla. So it's going to travel from the medulla then to the thalamus. So if you remember, again, back to our different sensations, things like hearing and vision, we have that crossover where the left side of the body is processed on the right side of the brain, and the right side of the body is processed on the left side of the brain. So that happens here as well. It is kind of interesting because not all neurons cross over, just most of them cross over. So touching an object can also stimulate the same side of the body as well, but not as intensely. I thought that was an interesting fact to include. And so, of course, our sense of touch is going to be projected up to our somatosensory cortex. And we've already looked at this area when we were looking at sort of the different parts of the brain and what they do. So this is our projection area for the body sense neurons. And if you remember, it's located in our parietal lobe, just behind the primary motor cortex, just behind that central sulcus. So this diagram would be back from chapter 3 when we were looking at all the different parts of the brain. And it's going to be the red strip. So it's called the postcentral gyrus, and then in brackets, the somatosensory cortex. So that's the part that we're looking at here. And we've already talked about the fact that our somatosensory cortex is set up in a fairly logical way, where adjacent parts of the body are typically located adjacent to one another across that cortex. So we could also call that a somatotrophic map, which is a representation of the body in the somatosensory cortex, where adjacent body parts are represented in adjacent parts of the cortex. So we had looked at, I think we called it a homunculus, where here we have the motor cortex and the somatosensory cortex, but sort of the thumb and fingers and hand are all sort of close to one another, both on our body and where they're processed in the brain. So we get this sort of mapping out of where things fit together. And it isn't just touch that does this. Within our visual centers, we actually respond to visual stimuli in the same way that they're laid out in the environment. So the way that stimuli respond or trigger our receptors on our retina actually lines up with stimulation in the brain. So it has that same, what did we call them? Somatotropic map. And we also have something similar with our audition, where we tend to have activation in the brain along a continuum that lines up pretty nicely with our cochlea. So going from sort of low frequency to high frequency, and that lines up in our brain as well, which is very cool. And so we're going to go into a little bit more detail with this somatosensory cortex, and we're going to break it down into our primary and secondary somatosensory cortex. And if you've done any of these different brain areas, you usually find that primary denotes something that's happening earlier on, and then secondary is where information that's been processed is going to be dealt with. So you sort of deal with the primary area first, and then the secondary, and that's what we're going to see here. So our primary somatosensory cortex consists of 4 different areas, and each is going to contain a somatotropic map of the body. So that's that layout that lines up with the different parts of our body. And it's going to play a role in processing sensory information from our body. And so the pathway that's involved here is, so that information is going to come in. It's going to go from our thalamus into the first two sub-areas. And they're sort of arranged in almost like a hierarchy. So there's a predictable order. So the thalamus is going to be sort of a raw, send it where it needs to be. Then the first two sub-areas are going to extract a little bit of information. And then once they've done that extraction, they're going to send that more refined information to the next two sub-areas. So it's kind of like we did a rough pass. Now we're going to go in more depth. We're going to pull out more useful information. We're going to do more with it and then keep passing it along. And so those other two sub-areas will process that information again in more depth, and then pass that on to our secondary somatosensory cortex. So we're moving along sort of in that hierarchy where we're getting more and more processing as time goes on. And so at this point, we're still going to have the information from the right and the left side of the body pretty much segregated. So the information from the right side of the body is mostly on the left, and the information from the left side of the body is mostly on the right. And again, not all of the neurons do the crossing over thing, but most, and we're going to treat it as most. All right. So we did our processing to a certain degree, and we've passed everything on to our secondary somatosensory cortex. Now our secondary somatosensory cortex is going to integrate the information from both sides of the body. So instead of keeping it separate for left and right, we're going to put it together so that we have a picture of what our body is doing as a whole. And we have special neurons in this area that are responsive to stimuli that have sort of an acquired meaning. So this would be certain orientations. They're going to be active if we're facing in a particular direction. Most of the research I've seen on this have been in rats, and they're sort of, I think they're called place cells. But basically, if the rat is in a particular location in their home cage, then this part of the brain will be active. And they have them to detect different orientations. So if they're facing sort of east versus 45 degrees angle, or a 45-degree angle over, they don't necessarily know that it's east versus whatever, but it's more of this is an axis in this cage, and if I'm turned off of that axis, then there are other cells that let me know how much my orientation has shifted, which is really interesting. And that's sort of up-and-coming research looking at, okay, well, what kinds of information is being told using these specialized neurons, and is it just in rats, or are there other species that have it as well? So we're getting more and more evidence in more and more species that this is a thing that just happens, which is, again, very cool. And then we're also going to have oh, okay. I was like, what was I trying to say here? So we're going to have connections between this somatosensory cortex and other parts of the brain. So we're going to start making connections with other areas, specifically the temporal lobe and our hippocampus. So when we talk about our hippocampus, we're usually talking about memory. So if we're having activation in our hippocampus, when this secondary somatosensory cortex is active, we're usually expecting some kind of formation of somatosensory memories. Basically, our ability to remember things isn't restricted to, say, factual information or events. We also have memories of actions that we've done and things that we've felt. So the hippocampus is probably being activated in this part because it's integrating sort of some of those things and doing memory formation for some of the things that we're experiencing. So lots of things coming together here. So our secondary cortex is doing all that integration and bringing information together. And then we can also have our posterior parietal cortex. And this is another association area. So we're going to be bringing things together. But here we're going to be sort of integrating our body senses as well as our vision and our audition. So we're getting all of our senses coming together. And if we look more in depth into vision and audition, you have primary and secondary visual cortices and primary and secondary audio cortices. So they're going to have that same hierarchy of processing. And then this posterior parietal cortex will take that more refined information from all of those senses and start combining it together. And it's going to allow us to integrate our body and our understanding of where we are into the rest of our world. So we're going to figure out where are we, what are we doing, and what's going on around us by integrating what our bodily senses are telling us, what our eyes are telling us, and what our ears are telling us. So bringing it all together. And so this would be the area that lets us know if our vestibular sense is disagreeing with our vision. So we can blame this for that spinning sensation when we've spun around on a chair. And so we have different sub areas that are going to be responsive to different sense modalities. And it seems that they're not solely perceptual, so there's other stuff going on here and other things that are being integrated, but again, not quite all that well understood. And we don't just see firing when we have an active sensory signal incoming. We also have almost like preemptive signals where neurons will fire while we're doing movement and while we're doing things, but also in advance of doing movement. So it's almost like we get a warning or a heads up signal that says, hey, we're going to move, be prepared for that. So it's kind of neat to see that it's not just sort of sensory information that's going on, but we also get to see some involvement in planning, which will integrate with other areas of the brain as well. So the deeper we look, the more complex it starts getting. And then for the last two things I wanted to talk about in this section are body integrity, identity disorder, and out-of-body experiences. So if we have this complex system that's set up to allow us to sense our body and what it's doing and how it moves and how it's interacting with things, of course there are going to be situations where things go wrong. And so a body integrity identity disorder, or apotemnophilia, is a condition where individuals that have no apparent brain damage, no sort of identifiable disorder, end up being convinced that one of their limbs doesn't actually belong to them. And so if you've watched any of those medical shows that come on TV, there's lots and lots of episodes I can think of with this, where individuals come in and they're convinced that that part of their body belongs to someone else, or it's dead, or it's an imposter. And it's because they don't have the same activation that tells them that it's part of themselves. So we have some damage to our systems of integrating all that information. And when they go into more detail, and when they study people who have this disorder, they find that when the limb is touched, there's no response in the parietal area that should be lighting up in response to that touch. But it's not that they're not getting any kind of sensation there. In fact, they see increased skin conductance in that limb, which actually tells us that there might be some kind of intense emotion. So they're having a response to it, but it's not the response they should be having, and it's leading them to have some kind of probably anxiety or sort of distress about the situation. And so again, another one of those disorders that isn't overly well understood, but does seem to be involved in some kind of damage to the systems we've been talking about. And then for out-of-body experiences, this is an illusion where an individual hallucinates seeing their body from another location. So sometimes if people are in sort of severe emotional situations or traumatic situations, they might dissociate and feel like they're looking down on themselves from a different angle. And so this can happen for natural reasons, but it can also be sort of intentionally stimulated. So if you use electrical stimulation, you can affect the parietal temporal junction. So again, we're involving that parietal area where we're integrating information. And there are, the textbook goes into more detail about it, but there are some really weird cases where stimulating individuals' brains in this particular area can actually lead them to seeing sort of duplicates of themselves. And so they go on to report it like, there's a exact double of me that stands just behind me that copies all of my actions. So it's kind of like they're seeing themselves in double, and they're trying to sort of wrap their minds around what it is that's being produced by these strange stimulations. And it can also happen naturally through things like epilepsy or brain injury. I guess natural is not quite the right term, but sort of without direct intentional intervention. So sometimes individuals will have that sense of seeing themselves doubled or mirrored, and it can kind of mess with their perception. All right. Okay. So our next topic is to look at pain and pain disorders. And so we spent quite a bit of time talking about different ways that we can perceive pain. And so we had sort of intense pressure or intense temperature. We can have tactile tissue damage. We can have chemical damage, all sorts of different kinds of pain signals. And as we said, it's going to begin with our free nerve endings that are going to respond to one of these types of stimuli. And the term that they use to describe what is produced by this kind of damage or pain is called inflammatory soup. So it's an array of signaling molecules that's released when the body is in pain. And so this is their very highly technical term for a mixture of things that includes histamine and different proteins and lipids and neurotransmitters and cytokines that are all going to be giving us sort of our bodily responses to pain. And so some of those things that are released are going to stimulate our pain receptors and let us know that we're in pain. But they also produce sort of the other side effects of being in pain. So if we have damage to part of our skin, maybe you get a paper cut. You're going to feel that pain, but you're also going to get a little bit of swelling and redness and inflammation as your body starts dealing with the fact that you've been injured. So that ends up enhancing the excitability of our pain neurons, so that, when you get a paper cut and now your hand hurts really badly and even the slightest touch is going to be too much, that's part of this response. And it can be annoying, but it can also be adaptive because it's letting us know that that part of our body is injured and we probably shouldn't be touching it. We should probably be letting it heal and not using it as much as possible. So it would lead us to start guarding that injured area. Maybe we're not going to be, I guess if it's on our hands, we have pain with our hands, maybe we're going to use that hand less, which is going to lead to less touching of things that might infect our hand. But again, as we said, the resulting pain can be really frustrating, but there is some kind of biological rationale behind why it exists. All right, and so when we're detecting pain, I think we had this diagram already when we were looking at sort of how sensory information is transmitted, but if not, we're just looking at the two distinct pain pathways. So we have a fast and a slow pathway. Our fast pathway is focusing in on our localized pain, and it's going to send information up to our cortex very, very quickly, hence the name. The slow pathway is going to be less localized, so it's going to tell us that this general area is in pain, but it's going to be longer lasting, it's going to move a lot more slowly, and it's going to give us that sort of dull, aching, or burning pain. And I always use the description of, when you stub your toe walking through your living room and you kick your coffee table. you have an immediate response of ow, and that's your fast pathway that tells your brain that you just kicked your table and this particular part of your foot is now sore. And you have just long enough to go, this is gonna hurt. And then you get that full body throbbing where your whole foot hurts and everything sort of aches and burns. And that distinction in those two sensations really accurately gives us the difference between our fast and slow pathways. One of them is going to happen really quickly, one of them is going to take its time, and they're going to give us different information. So fast is exactly where it's hurting, pretty much as soon as it happens. And then the slow is longer term. This area is still sore. Maybe take care of it or avoid it. And so our slow pathway is going to depend on very thin and unmyelinated neurons. So these are our C fibers. And so if we remember back to how our neurons work, myelination allows signals to jump ahead. So having very thin and unmyelinated neurons means that every single part of that neuron is going to have to fire to pass on an action potential, hence being slow. So these C fibers are going to take a very long time for that information to pass along. And then the fast pathway is going to be controlled by thicker and myelinated neurons, and we can call those our A delta fibers. And so those are going to be surrounded in that myelination, which allows the signal to travel faster, which is kind of useful. Okay, and so if we think about it, those very dense A delta fibers for our fast pathway are going to tell us exactly where and pretty much exactly when we've been injured. And it's not going to persist. Once we know that we've been hurt, it's done its job. But the C fibers, they're typically located a little bit deeper in the skin surface, and they're giving us that aching and enduring pain. And it basically just reminds us that we've been injured and that we should be protecting that area so that it doesn't make it worse. And then when these signals enter into the brain, we can send them off to many different parts of the cortex, as well as we can send them into subcortical areas. So the cortex meaning like the surface, where we have things like our somatosensory cortex. But we can also send it deeper into the brain, those subcortical areas towards the middle, and that would be things like our amygdala or our hypothalamus. So if we're dealing with pain that's going to lead to an emotion, that's going to be activating the deeper parts of the brain, specifically the amygdala. And so depending on the nature of the pain and the situation that we're in, we'll see different activations at different levels of the brain. All right. And so we're going to talk very, very briefly about some of the substances that are released and help us help us experience pain is not a great way to say it, but they're involved in us experiencing pain. So we have a couple of neuropeptides, and these basically serve to enhance a neurotransmitter's effect at a synapse. So substance P is a neuropeptide that increases pain sensitivity. So this would be part of that inflammatory soup that happens when we're injured. And so substance P is basically going to make those neurons more receptive to stimulation. Those free nerve endings are going to be more receptive to stimulation so that we can feel that pain more easily. And as we said, feeling that pain leads us to protect the area, which is good. We also have glutamate and substance P. So glutamate would be for mild pain, but substance P would be for more intense and enduring pain. So early on, we'd have glutamate released to help us feel that pain, but if it's bad enough, then we would also get the substance P to have that increased sensitivity. But they're both going to be released in our spinal cord, and both are going to lead to enhanced sensitivity. They just differ on when they're released. So there's sort of a threshold of how intense is this pain? Are we having just glutamate, or are we having both? All right. And then, of course, we run into this issue where, okay, how do we treat pain? How do we prevent the feelings of pain? And we have quite a few different kinds of general anesthetics. So these are substances that would render a patient unconscious and therefore unable to feel pain. But as with a lot of things that deal with sort of consciousness and regulations thereof, the mechanisms aren't very well understood. So we have sort of different drugs and drug classes. So we can have non-steroid anti-inflammatories, we can have opiates and acetaminophen, all of these different things. But the methods of action aren't necessarily precisely understood. So this diagram does a good job of sort of what vague idea is it doing. So our NSAIDs, they decrease swelling in the tissue. So they would decrease that inflammatory effect of our inflammatory soup. Same with our COX-2 inhibitors. So they're going to be working on decreased inflammation. And acetaminophen is an interesting one because it blocks pain signals in our central nervous system. Again, very vague. They don't talk about the mechanism of how that happens, mostly because they can't quite agree on it. And then opiates are going to actually do the opposite. So instead of blocking things like that, well, I guess they're going to end up blocking eventually, but they're doing so by activating opiate receptors. So these are going to respond to things like, or naturally they'd respond to things like endorphins, which would help us feel less pain by inhibiting those signals. So here the opiates are taking on that role, hence the name. All right. Oh, good. And I did think about endorphins. So endorphins, they're basically a weird category of chemicals in the brain, and they can function as both neurotransmitters and hormones. So there's sort of this distinction between neurotransmitters and hormones, where hormones are typically passed through the blood, and neurotransmitters would be more at the level of the synapse. But that's not necessarily as hard of a distinction as it has previously been thought. So we can have sort of, they're neurotransmitters and they're hormones. They can act locally and more broadly. But these endorphins, as I said, are going to act at those opiate receptors within our nervous system. And they only function under very, very specific conditions. So we will release endorphins if we're in a situation that we need to not feel pain. Because before we were saying that, feeling pain can actually be beneficial. It leads us to guard an injured area. It can lead us to be more cautious and avoid further injury. It can be beneficial. But in some situations, being in pain is going to be detrimental. So if we think of, say you're getting attacked by a bear, yes, you have a bite, and it is probably important that you know that you were bitten, but constantly being reminded that you were bit while trying to fight off a bear is not really your best course of action. So your body could inhibit that signal of pain so that you can focus on other things like getting away from the bear. And so inescapable pain is more likely to release endorphins. And what this means is if it's a situation where you're going to continually be in pain and there's no way to stop it, you're going to produce more endorphins to sort of nullify those effects. So your body almost recognizes that there's no benefit to staying in pain, so it's going to release endorphins to get rid of that sensation. And we can have a substance called naloxone, and it blocks our opiate receptors, so it would basically counteract our endorphins. It also counteracts opiates, which is what it's usually used for, but we can use it to eliminate that analgesia, or the sort of lack of pain sensation that comes from our endorphins. And so they've used the sort of functionality of naloxone in order to sort of figure out, okay, is this lack of pain or is this reduced pain because of endorphins, or is it because of something else? And we only see an increase in pain in situations that are sort of more intense. So specifically, inescapable pain, that will be, or that's something that we're pretty confident is related to endorphin functioning. But escapable pain, so something that you can avoid, something that you can prevent, that tends to have less of an effect, which tells us that escapable pain is probably mediated by a different system. And that kind of makes sense because it's less intense and less sort of full numbing than intensive situations. But it's kind of neat to look at some of the research in that area where they can specifically look at, okay, well, what happens if we give naloxone? Does it bring the pain back or not, and to what extent? So some very intelligent studies have been done with that. And we can also have other things that result in endorphin release. So physical stress, so actual physical sensations of the body, you can stimulate their release through acupuncture. Sugary food consumption also releases endorphins, so it makes us feel good and therefore feel less pain, which is one of the reasons why we tend to eat so much sugary food, because it makes us feel better. So you've probably used the term comfort food, and this is why, because it does actually provide physical comfort, which is very interesting. And endorphins are also released by sexual stimulation. And I found it interesting, the textbook goes out of their way to specify that sexual stimulation in rats and humans causes the release of endorphins. And I understand that that's because they were looking at two distinct studies, one for rats and one for humans. But I was like, are you intentionally excluding other animals? Or it was just a very strange wording. But yeah, okay. Okay, and so that's how our endorphins kind of work in our, again, not super well understood method. Our next vague topic is to talk about how we inhibit pain circuit. So how do we actually block those incoming pain signals? And the most common theory that addresses this is the gate control theory. And this is basically saying that signals arriving in the brain will trigger some kind of feedback, an inhibitory message that will travel back down into the spinal cord, where it closes off some kind of neural quote-unquote gate in the pain pathway. So if we think of all of those sensations coming in through the spinal cord and then up to the brain where we can interpret them, if the pain is intense, if it's something that we need to block out, we can send a signal back down that says, stop sending these signals, we don't want it. And this is kind of a weird one to think about, because when they first proposed it, they thought there might be like a physical structure, something that interferes with those signals being passed up the spinal cord. But despite all the research that they've done, they haven't been able to find sort of a physical smoking gun, as it were, of what's causing this. So when we're talking about a gate in that pain pathway, the best way to try and envision it is that we're referring to a pattern of neuronal firing that seems to sort of interfere with the transfer of those sensations. And that's a very vague description because we don't really know what's going on. So we have had quite a bit of research that lines up with this general idea behind the theory. So gate control theory seems to be supported by many, many studies, but we are still completely at a loss as to what mechanism is driving it. So we are fairly certain that, yeah, there's probably some kind of descending pathway in the spinal cord, and where it's probably going to be involving the release of endorphins to control pain. So if we experience enough pain, then we're going to send down a signal that triggers the release of endorphins that would then reduce our perception of that pain because we'd have less responding. And that's about as much as we are fairly certain of. And so they think maybe this would involve the periaquidrial gray, or the PAG, and this is just a part of our brainstem that's going to surround some of our cerebral ventricles. So if you remember, our ventricles are those almost channels of fluid through the brain. So they're going to have a large number of endorphin synapses. So in that area, there's lots of parts of the brain that could release endorphins, and specifically in and around those ventricles where the signals could be transferred around. So that's why they think that area might be involved. And we do know that endorphin release ends up inhibiting the release of our substance P. So substance P increases our sensitivity. So releasing endorphins, inhibiting substance P, is going to then sort of reduce our sensitivity again. And it might go to sort of normal levels, or it might go even further into we have reduced sensitivity as per our average. So it's sort of vague still. And then activation of this endorphin circuit is going to be sort of triggered by multiple areas. So just to add to our complexity, we can see the cingulate cortex triggering this release of our endorphins, especially if we're looking at placebo analgesia. So if we tell someone that something is a painkiller, we do see a decrease in their pain response, but not because of whatever was in their sugar pill. It's because of the fact that they think it's going to work. So the cingulate cortex seems to be involved in that process. If we stop feeling pain because we're experiencing extreme fear, that's usually initiated by the amygdala. So anything to do with fear is usually the amygdala. There's a bunch of different potential triggers. And so these sort of multitude of different starting points that all activate the same pathway is a large part of why there's so much confusion about what's going on here. All right, and then the last two topics for this section are talking about the two extremes of pain. So congenital analgesia is basically an insensitivity to pain. And again, watching medical dramas, there's always a good episode that deals with someone who feels less pain. I fairly recently watched one of the Grey's Anatomy one where they have a little girl who doesn't feel pain. And it does a really good idea, or a really good job of displaying that it can be seen as beneficial. The little girl thinks it's a superpower, that she can't be hurt. But it's also fairly dangerous. So if you think about it, someone who doesn't feel pain doesn't get those signals that lets them know that they're being damaged. They don't know that they should be protecting an area. And it can lead to injuries that go untreated and can cause problems and even death. So they can injure themselves, but they can also injure others because they don't have the concept of others experiencing pain because they can't themselves. And there's a bunch of different mutations that seem to be linked to this particular disorder. And we can also see that some of them might be due to elevated natural opioid levels. So if they have lots and lots of endorphins naturally present, they would have a reduced pain response all the time. So we can kind of see that going on, but there's lots of potential sources here. Our other extreme is chronic pain. So instead of never feeling pain, some people always feel pain. And so when we define chronic pain, this could be pain that persists after healing has occurred, or that is occurring beyond the time in which healing would be expected to occur. And they include that because if they're still feeling pain, theoretically it's not fixed, but we would have expected them to be back to normal functioning, and they aren't. And we have lots of interesting correlations here. So depression is strongly correlated to chronic pain. And that's an interesting one. With depression, that sort of negative state of mind actually makes negative emotions and feelings like pain more intense. So your internal state can actually increase what you're sensing there. And so there's sort of a vicious cycle where individuals who are depressed tend to feel more pain, and feeling more pain tends to make them feel more depressed. So you can actually have a spiral where they sort of feed back into one another and run out of control. We also see some association between the functional connectivity between the nucleus accumbens and the frontal cortex. So the more of connection that we see between those two structures tells us that we will have more chronic pain. And this was specifically from a study looking at back pain, where if they could quantify the sort of co-activation of those two areas, they knew which individuals would experience pain a year later. And they had, I think it was an 85% accuracy in this prediction, which for these kinds of correlational studies is massive. So this gives us the idea that maybe there's some kind of emotional and or stress component involved in chronic pain. So if somebody has high activation of areas associated with emotion, or if they are very prone to stress, they might also be prone to this chronic pain. And of course, again, there are some genetic studies that have identified particular genes that tend to have mutations more in people who experience chronic pain than those who do not. And so one of them is the same gene as the one that's linked to insensitivity to pain, but it's a different version. So typically we think of having our normal wild type allele, the standard version of a gene, and then you have an allele that's non-functional, and that would be our mutation. But you can also have mutations that are maybe over-functional. So maybe they do something different or more extreme than the normal. And so that's what's going on here, is where we have different alleles that can lead to either oversensitivity or undersensitivity depending. But there's also a lot of other genes that might be involved as well. All right. Oh, and these are just some of the areas that we've been talking about, again, including the visuals for those who are visual learners. But if this helps, so we have that PGA is shown with that red dot. And then this is just sort of showing the movement sort of from our surface more deep into, say, our brainstem, which is then going to trigger the release of endorphins and going to affect the responding of our substance P. Okay, I think I'm just going to finish up with this slide because we still have, this is to do with chronic pain. This is just looking at sort of our long-term changes in the nervous system because of chronic pain. So we do see both functional and structural changes within our bodies if we experience chronic pain. And so it kind of makes sense. As certain pathways are used more, those pathways tend to be strengthened. We see sort of a stronger connection between areas that tend to fire together. And so as these pain pathways are activated more and more, they become even more sensitive because they're forming stronger connections. So because they're used a lot, they then become more sensitive, which means that they're used even more. So again, that sort of positive feedback spiral. We also see new connections between peripheral neurons in our spinal cord. So we start having sort of the neurons that are coming into our central nervous system. They're going to start forming connections in the spinal cord. And we also have normal spinal inhibitory mechanisms end up depressed. So we can see sort of a reduction in our ability to inhibit pain signals as we experience them more. So as people endure chronic pain for longer, the symptoms can actually get worse, not because whatever's causing the chronic pain is getting worse, but because their body is actually changing due to the experience. And within the brain itself, we can see lots of changes due to chronic pain. Again, those brainstem pathways, those ones going here through the brainstem that are started with that PGA, those are going to become more responsive because they're being more activated. So more connections, because they're used more often, leads to more use and leads to more connections. We also see increased activation in the prefrontal cortex, the anterior cingulate cortex, and the insula. So again, feelings or things associated with feelings and emotions are going to be activated as well, which kind of makes sense. If you're in pain, it's going to trigger the part of your brain that responds to pain. We end up seeing more of our somatosensory cortex devoted to painful areas. And that kind of makes sense. The more we use a part of our body, the more it's going to be represented in the brain. So if you have chronic wrist pain, the part of your somatosensory cortex that shows your wrists is going to get proportionally larger. So we'll start seeing more connections there, so it's going to have a larger volume, which then leads to more sensation of the pain that's there. And we actually start seeing a loss of gray matter as we have more and longer experiences of pain. So we would have sort of less and less neurons in general, but the ones that are there are going to be producing more connections. So we'll have more of the white matter and less of the gray matter, which is very interesting. Oh, I did only have one more slide. Okay, I'm going to do this one really, really quickly. So phantom pain is another experience of feeling pain, but this time it's specifically pain experienced in a missing limb. So if somebody has an arm or a leg amputated, they sometimes still feel pain where their arm or leg would have been. And it's not necessarily that they're feeling pain at the amputation site. It's not that we've severed neurons and that severing is causing pain. It's actually that they're feeling pain sort of where the limb should have been, sort of past the amputation point. And so they think that this is due to the presence of cortical areas that are still devoted to the missing body parts. So you had sort of an equal representation of your right and your left arm. And if you no longer have your right arm, you still have the parts of your brain that should be sort of receiving sensation for that, but they're not being stimulated. So they might be stimulated by something else that's causing you to interpret it as pain in the thing that is no longer there. And interestingly, anesthetics typically don't relieve phantom limb pain. So it's probably not from an input of those nerves that were severed, it's probably something else. And again, a bunch of different theories. They say maybe there are foreign neurons, so things from other parts of the body that have kind of encroached on that area because it's no longer being used, but we're having sort of cross-firing where the presence is triggering us to feel something from that area. But it could also be because we have sort of a reduction in signaling in that area now because we don't have any input. And so other things that were present in the background but that we never experienced are now coming to the forefront. So they were pre-existing stimuli that weren't usually perceived, but now that nothing else is going on, they are. And again, not quite sure what the cause is, and it might be different between individuals, but those are two pretty good sort of propositions for explaining that. Okay, and that is everything I had prepared for today. I didn't think I was going to get it to the end, but we did. And then, as I said, we're going to pick up with movement on Thursday.